So I was given the topic of trapping because we're doing a lot of work in trapping in our lab, and I think it's important to catch you up with what we've been doing. You'll see on the last slide that the first name on this presentation is Danielle Kirkpatrick and not Larry Goot, because this is all of her research for her PhD, and Danielle's back there sitting next to Heather. So you're really going to see her work here. And this first slide is just a little review to catch you up with where we've been. So we started out, the primary trap that everybody's using is one of these cup style traps down at the bottom, and they bait it with a commercial lure, and the Sentry lure, the one by the company Sentry, is the one most people use. Or they've bait it with this sugar yeast solution. Her work has been trying to look at different kind of, we don't think these are very efficient traps. There's a lot of flies that don't go into them. There's a lot of flies that get out once they get there. So we wanted to go more toward what the industry has done for many other insects, and that's some sort of sticky trap where once the insect gets there, it doesn't go anywhere, it's stuck. So you don't lose a lot of the flies that are actually attracted. So she showed in the last year or two that you could, she showed a couple things. One thing she showed is red was really attractive to SWD. So you had some red component to your trapping. It really increased the number of flies that came. And then she showed that either a sphere or a panel, red panel or a sphere that was colored red were very attractive traps. And you can see in this data that the sphere with the Sentry or the panel with the Sentry, which are the two bars on the left, are much higher than the bars on the right, just showing that you're going to catch a lot more flies with these traps. So we wanted to go out in the field this year with these traps and actually confirm that it's really the red panel we want to go toward, because there's several companies out there that are selling yellow panels for SWD and I think white panels for SWD, sticky traps. We set up a study that we ran down south, and then Nikki's group ran it up here with these different colored traps, and we had a cup trap baited with the Sentry lure as a positive control. And then she also had a panel trap that was a yellow panel with a red sphere, kind of a combination looking at, we had some lab data that showed a contrast was really important to SW as well, and maybe that would be even better. So ran this trial at I think 10 or 12 different sites across Michigan, and the red panel and the combo trap looked like they caught more flies than the other traps. The other colors, not statistically different, except for the yellow panel is by far the least effective at attracting and catching flies among possibilities for a panel trap. The panel trap, for the most part that people were using this trap, and I interacted with a lot of people in the field, did not like the red panel trap. We don't have a commercial red panel trap. This is just a panel that we had a company make for us. And when you try to identify the females on those panel traps, it gets very difficult because you have to look at their egg-laying device, their ovipositor, and it's in the stickum, and it's a little bit hard to do. The males here shown on the right are pretty easy to identify in this. You can see the spots on the males, but the females are kind of a pain. And plus the stickum was kind of a pain in that the lure would get caught in the stickum when it's blowing around in the wind, and there were some problems with it. But one of the things we were interested in doing this on a larger scale, and why we incorporated it into the trapping network, is we had the idea that maybe you don't even have to count females. Maybe you can just count males, the easy ones to identify, and is it really that important to be counting females? And I think the jury's still out on this one, but if you look at this is all the sites from the northwest down to the south. You look at the red panel trap, and you can see that the pattern is pretty much the same for the females and the males. And with the cup trap, the pattern is the same as well, except maybe you catch a few more females early in the year with the cup trap that we weren't catching on the panel trap. But I think, to me, there's evidence in this data so far that maybe you don't even need to count the females, because back here where you're catching these first few females, I don't think it's telling you a lot about, the fruit aren't ready to be infested at that point. By the time they are, I think the panel just catching males is probably doing a pretty good job, but the verdict is still out on that one. And also I wanted to say that I'm working very closely with the Tracy company right now to try to develop a red panel trap that is more user-friendly. You have the yellow panel trap that you use in your cherry fruit fly monitoring program, where the two panels come apart, the stickum's in the center, there's no stickum getting all over it. the stickum's only around the central part, so you have a place to put your lure. So they're going to make us a nice red panel trap. They're actually making 5 or 6 different versions for us that we're testing in the greenhouse by releasing flies now to see if we can come up with the most user-friendly version of a red panel trap. Danielle also did a second-year study to look at, well, in what area are these traps actually bringing flies in? And if you put a trap out, what is the area you're actually monitoring? To do this, she did a mark release recapture study for SWD. And in this study, what she does is she puts a trap in the center of a very large cherry orchard, and then she marks flies with fluorescent dyes shown here on the bottom. So you mark flies different colors, and the flies from each distance are marked a different color. Then she releases them from all 4 cardinal directions, catches them on the trap, and then she brings the traps back in. You can see this trap at the bottom that's lit up. So you put it under UV light, and you can see the flies glow different colors depending on the distance they came from. When you take that distance data and plot it in a graph without transforming the data, you get a curve that looks like this. And we know if you get a curve that looks like this, that has this asymptotic approach to 0, and it almost gets to 0. You have a nice curve that looks like this. It means you pick some distances that are very good for the study. They're giving you very solid information. You got to the point where you're probably to the maximum distance for most of the flies, so you have a good data set. She catches back a little over 1%, which is typically what we've been finding for every insect we work with, whether it's codling moth, oriental fruit moth, leaf rollers, Japanese beetle. When we do these studies, your trap is catching about 1 to 2% of what's out there. When you transform that data and get this shaped curve, where that curve crosses the axis, that's the maximum dispersal distance for 95% of the released flies. So 95% of the flies that you release for SWD traveled a maximum of 90 meters over the course of this trapping experiment for 1 week. So their maximum dispersive distance, we call it, is 90 meters. That's actually 95% of the population. If you carry out the calculations for what that means for you when you're trapping your orchard, we know now that at least for the red panel trap with a Sentry lure baited, that trap is monitoring an area of about 6.7 acres. So those flies are coming from an area of about 6.7 acres. Majority of the flies are probably coming from a couple acres away, but all the flies, all told, are coming from about 6.7 acres. The plume reach from that, the actual amount of volatile plume that's, once the flies get into that plume, it's only about 3 meters. So most of the flies that are coming to your trap are randomly getting there, and they get in your trap. They only are going to get in your trap once they get to about 3 meters, then they're going to make it to your trap. If you look at the numbers, Danielle, if you look at the catching 1.1%, if you translate to the population out there, if you catch 1 fly in a trap for 1 acre, your population per acre is about 29 SWD. If you're catching 10, it's 290, and then it's close to 3,000 if you're catching 100 of them in your trap in that 1 acre area. So that's really my trapping talk, which would have done a really nice job of only taking about 7 or 8 minutes, maybe 10, and that would have been good. But as I go around hearing about the research that we're doing, what I hear from the extension educators and I'm hearing from growers are basically these 2 questions. Is trapping worth the effort? Do we even need to do this? This is kind of a waste of time. And then follows from that, why should we continue to even look for better traps and attractants? And that's kind of a sad question. I put Danielle's picture in here because if it turns out that we shouldn't, then Danielle's wasted 3.5 years of her poor life because her PhD is entirely trying to figure out trapping. So I wanted to not just let that go around in the air and let everybody think about it. So I wanted to directly sort of talk about those 2 things for 5 or 10 minutes as well in this presentation, because I think it's a really good question. They're both really good questions, important ones to think about in a think tank group like this. And really what this is about is thinking about questions like that. So let's try to talk about that a little bit. So the first thing I wanted to put up there is, remember, we've been trapping SWD for a number of years now, and in some years, in some regions, SWD may not have been present until very near or after harvest. So if you look at this first figure, for example, the lucky people in the Southwest for most years until this year, it would have been really important to trap. This information was critical for them because basically they're catching no flies all through tart cherry harvest when their fruit are susceptible. So they got away with maybe one spray. They knew that they didn't have any flies, they didn't have to worry about it. And trapping was pretty critical to them. In the Northwest, not the case. There were maybe early when we first started, you might have seen that, but the last 3 or 4 years, it's pretty much, you're starting to catch flies at cherry harvest. And maybe in cherries in the north and in blueberries, it's not so clear that trapping is really critical. The critical thing probably is having a model that tells us when the fruit are susceptible. And if you were to say, okay, it's going to be like this every year in the north, and maybe it's every year in the south like it was this year, the answer to the question is probably trapping's not all that critical. It's really not the most important information. But I don't think that's the case. I think in the south, we're still going to have situations where trapping is very critical, and in west central Michigan, and you're going to want to know if the flies are there or not, and they might not show up until very close to harvest, and that trapping information is very important. For the poor people up north, Nikki and her group, they may have a strong case of saying, wow, trapping's a lot of work, and we're going to need to treat every year. I don't know the answer to that. I still think that you need to know that every year, how intensive it is. So that sort of directly answers that. The other part of this that we're kind of ignoring still, I think, and Danielle and Heather and others have been working on this, you won't see too much about it, but we're trying to learn about it, is we still don't know where they are and how many are out there in the winter and early spring. So we're only trapping this fly successfully once it's getting close to harvest and close to time to make decisions in the summer. But working on trapping to try to catch these overwintering flies, I think, is really important. We still have not maximized the kind of information that we can get out of trapping. We're working really hard to get there. So it's just a reminder here that it's not all about trapping these summer morphs. What the heck are they doing in the winter? And it would really be nice to know. So we've been starting to work on this. This is what the flight of SWDs look like so far in Michigan when we trap for the whole year. You basically catch a lot of flies. You see us peaking here in August, September-ish. They crash down, and we start to catch overwintering flies down here. But basically, if you have traps out there January, February, March, April, where the heck are they? We don't catch any. It'd be really nice to have a trap that catches them then, or maybe there's none there. Who knows? So trying to understand that part of trapping, I think, is still really critical. This is what it looks like. Danielle summarized all this data from last year, looking at September. They're all summer morphs, and then we have this winter morph that is much bigger and survives better in the winter, and it starts showing up in October. About half the flies are winter morphs. Then by December, they're all this winter morph that's not doing any feeding. It's just spending the winter, and when it gets warm enough, they might be active. And then as it starts to get to springtime, they're doing something that we don't know, and we don't know how many are there. So Danielle and Heather have been working in the Kirdong lab trying to figure this out. And one of the things they've been trying to do is, do they respond to different volatiles? Maybe the traps we have are kind of worthless for these winter morphs, and we need a different kind of trap. So they set out starting to do this last year. The first thing they decided to do is find out what SWD responds to. Does the winter morph respond differently than the summer morph? And what they found, they did these things called electroantennograms, or EAGs. So they get in Kirdong's lab with this specialized equipment, and under a microscope, they capture these flies into micropipettes to hold them still. They put tiny electrodes on their antenna of these flies, and then they measure the response the fly has to different volatiles. When they do that, and they tested several different volatiles, they tested some that are known to be attractive to fruit flies, like acetic acid, banana flavor, methionol. And then they tested some that are a little odd, geosmin, which is kind of an earthy smell from a bacterial organism in the soil, and then a pine smell because there's people showing a lot of these SWD end up in the forest in the wintertime, and then a floral compound. When they did that, what they found is that if you look at the red, that's the response of the summer morph, and the higher response is the higher bar, and then the winter morph is blue. You see across all the compounds, the winter morphs in general don't respond as much to various volatiles in general. And then two of the compounds, geosmin and isoamyl acetate, there was a greater response. There was a greater response in the summer morphs than the winter morphs. This one's interesting in that this is actually maybe repellent to these and not repellent to our winter morph. So they're doing behavioral assays now to try to figure out what that means behaviorally, and we don't have any data on that yet, but just interesting to see. And then finally, there's a whole effort that's not just trapping, but associated with trapping is trying to find better attractants, or as Juliana mentioned, maybe repellents even for SWC. So trapping studies are not all about just trapping. They're also about developing attractants to do other stuff, which I think is really important. For example, insecticidal baits or developing an attractant kill for this insect require doing research on what they're attracted to, or you can't do any of that stuff. So I have a postdoc in the lab, Joanne Wang, who's working with yeast strains. She got yeast strains. UC Davis has a whole library of different yeast strains that you can obtain. She got a whole group of different yeast strains, two of which are known to be fairly specific to spotted wing Drosophila, and then she multiplies, basically raises those yeast strains in the lab to develop, to get enough material to do bioassays with them to see how attractive SWD is to these different yeast strains. This H. uvarum is what in this choice test where flies chose either the H. uvarum on the agar that the yeast is being raised on, or just the agar alone with no yeast. You can see it looked like this one that's specific to SWD was pretty attractive. At least a lot of flies went to that particular plate. And then also this one right here was pretty attractive. So after that, she went to the field in several different environments, and this is a cherry orchard in particular, and started to see it looked like H. uvarum would catch a few more flies than the others and had some potential in it. But unfortunately, the bad news is that she kept a positive control in there. So this is the Sentry lure, and the Sentry lure still wiped out all these yeast strains in terms of attraction to the trap. The important thing for this yeast strains, I think, though, is not so much for trapping, but rather maybe they're more important as an insecticidal bait, a very short range thing for the fly to respond to and not long range. And I think understanding the way they respond to these yeast is still really important. And we're looking at fruit volatiles and other kinds of things, but the point here was just that it's really important to keep working on these attractants, I think. So finally, the last thing is, one of the important things for the attractants is maybe you can come up with an attractant to kill. I think our group's working with about 4 different attractant kills right now. I'm sorry. And so we're working with our own particular version of attractant kill. We have one from another commercial company. Juliana referred to the splat one, and then we have this particular one. And I'm just going to show you quickly data from that last one because that one turned out to be the best of the ones we've done this year, just to give you an idea how effective they might be. So this is in large commercial cherry orchards, replicated 3 times, I think, or maybe 4. We put up this attractant kill device, which is a bag that holds the blend that's in the Sentry lure inside as a liquid, and there's a lot of it, so it's really attractive. It has holes so the flies can go in and drown, and then the whole bag is impregnated with deltamethrin to kill the flies that come and don't go inside. If you put them up at 210 or 70 per acre, this is the catch of SWD in the center of our 1-acre plots. So you can see at the highest number of these attractant kill stations, we actually had a dramatic reduction in the population. And this just shows you the total number we captured. Really had a dramatic impact on the population. I really think there's some promise for attractant kill for this SWD still. So that's all I have, Bob. I think I did pretty good, actually. I just wanted to finish with the thanks to Giuliani. Already mentioned the big team that's doing all the trapping work. We had funding for this trapping projects from Cherry Committee, Hort Society, Project Green, and from the two big grants that they talked about, the SCREE grant and the ROAR grant, and actually the MDAR block grant. And we had a lot of people helping us in my lab, technicians and postdocs, and then Nikki's lab always partners with us on all these projects, which is nice. And we've been working with Gerdong's lab because she has some really specialized equipment that makes this behavioral work interesting. And we had a whole slew of summer assistants helping us, some of which are shown here, having a great time.